My association with the Linnean Society started in about the early 1980s. This is me and Gordon Howes at the Natural History Museum in London in December 1982. I'll let you figure out who's who. This is taken in the Spirit building, which has since been demolished. I think it's the Darwin Center, or at least part of the Darwin Center is in its place, and a new facility. We called it the BM&H back then. We certainly still use that abbreviation for catalog lots, and I always think of it as the BM&H, only reluctantly call it the Natural History Museum. Well, anyway, Gordon Howes was a great mentor to a lot of ichthyologists who came through the Natural History Museum at that time. And I went there on a NATO-NSF postdoctoral fellowship, which was a spectacular program. And I know that Sandy Knapp also went over to the UK, and Dennis Stevenson. And if there's anybody else here who benefits from that program, let me know. I mean, it was really quite remarkable. Well, this seems like a long, long time ago. Certainly a lot of pigment has washed down the drain since then. I have been at the National Museum of Natural History of the Smithsonian for the last 27 years, and a very proud member of that staff. And all staff at the Natural History Museum will talk very eloquently and enthusiastically about the collections. And you've probably seen photographs like this before. This is one in the bird collection with all the drawers pulled out. And standing in the middle there is Roxy Laybourne, an ornithologist who developed a method to identify fragments of bird feathers using morphological characters, and her work continues through the work of Carla Dove, appropriately named, who was one of her students. Well, these photographs are so effective and so compelling. Everybody wants to do one. So here's one from the Department of Invertebrate Zoology, and of course they have some of their spectacular collections on view. It's interesting that the staff seems kind of dwarfed by these, but I think there's something appropriate about that. Entomology, of course, had to do it too, and of course we've seen some lovely butterflies already today, and here's some more. And well, mammals did it too, but it's a little different effect. But we like them nonetheless, as some people have mentioned. So very, very, very important part of the collections. Well, these photographs, we bring them out. We bring them out to say how proud and happy we are about the collections and how well maintained they are. I mean, look at that, what effort had gone into preparing all these specimens and laying them out and collating them and so on. But recently, and this was just on April 1st this year, I don't know whether it was meant to be a joke or not, but there was this posting on Imgur.com. I think this is probably an aggregator called Inside the Specimen Collections of the Smithsonian's Museum of Natural History. Was posted by somebody named Mike Pants, and I didn't investigate that at all. So what was funny is that on this post, and I think this also ran in the Daily Mail at the same time, or something similar on one of the Daily Mail websites in the UK. People, of course, respond to this. I mean, that's what you do when you post pictures. People have to say something. And these are some select positive comments. Now, this is not a random controlled test to get comments back about the collections. It's just I was pulling out the ones that I thought were appropriate. So here's some selective positive comments. As an archivist, I approve their beautiful organization. This is seriously interesting and cool. Thanks, Imgur, you rock. Utterly fascinating. Thank you for the post. This is hauntingly beautiful. So we all read this and said, yeah, they get it. They have no problem. We're doing fine. We can go back to work. But as you might imagine, there were negative comments, and they made our skin crawl. Never knew that the Museum of Natural History had such a hoarding problem. Damn hoarders. Someone needs to hire a good closet organizer. Does it spark joy? No. Out. This one was really disturbing. Good thing the Smithsonian's not in California with our earthquakes. Now here's somebody from California who doesn't know that there are natural history collections in San Francisco, Berkeley, San Diego, Santa Barbara, Los Angeles, and so on. And that's, I mean, we laugh, but it's the problem. Then the last one, and again, this is one of many. I selected these for discussion. That's ridiculous with how many they have. There's no way they could possibly go through all of those birds and insects. It's a shame. And so what this says to me is, we know that the public doesn't always get it when we have collections, when we bring them around and say, isn't this a wonderful thing? And just having it out there for public commentary, there were a lot more negative comments, and I think I didn't go through them all. I think the negative outweighed the positive, and some of them were just sort of funny and flippant, but these were, it's a shame, really hit home. And so I think, in American vernacular, we say, Houston, we have a problem. And what this means is the onus is on us. I can't, blame the public. I can't say, oh, you don't get it, because all they can get is what we tell them. And so what I want to talk about is, say, 3 ways at the Natural History Museum in Washington in which we are trying to bring the specimens out more to the public, have more members of the public use them, have other scientists use them, and how we can increase the kinds of collections that we keep. Now, these are just 3 activities that I'm involved in. That's inviting artists to study natural history specimens, mining old collections for new data, and to cryopreserve living cells. There are many other that people are involved in, other efforts that people are involved in our museum and other museums as well. But I just wanted to talk to you about some of these today. Well, bringing artists into the collection. In 2007, the Smithsonian began a program called Smithsonian Artist Research Fellowship, or SARF, and it was conceived by a fellow named Ned Rifkin, who's a former administrator of the Smithsonian, and he wanted to have artist fellows come into the museums. And these are not botanical illustrators or wildlife artists and so on. Those people have always come into museums and always been a part of natural history. But these are fine artists, visual artists, and so on, who could come and study with researchers and be inspired and develop their own artwork after being associated with the museum. And a woman, Susan Talbot, was the first director of the program. And so this is a relatively new program. And when it started, there's a fellow CJ Huang, as he was called. He's an artist who's originally from Taiwan and is now based in New York. And CJ makes kinetic sculptures, and he was very interested in bioluminescence. We heard a little bit about bioluminescence just right before the lunch break. And you can see here he's putting together one of his this is, again, one of his kinetic sculptures. He uses a lot of found objects or discarded objects. So these are just water bottles that have been discarded. This sculpture will move. I'm sorry, I don't have any films here of his work, but I'll give you his website at the end. You can go to that if you're interested to see some of them. The fluid in the bottles is the ink from highlighter pens mixed with water, and the whole thing moves he gets old discarded computer fans and puts them around these, and then just programs a sort of a little symphony, and they all move together in either unison or not. And I'll show you what a big one of them looks like. Well, anyway, CJ called me. He was one of the first fellows who had been invited to apply to the program. Initially, you had to be invited rather than just apply. And these are really, so most of the artists are well known. They're certainly well established. CJ has been at the Venice Biennale, for example. They're not people who just put around their garage on the weekends. I mean, these are professional artists. So he sent me an email and said, he was explaining what he did and that he was applying to this program. He had been asked to apply to the program, and he wanted to come to the museum, and he was interested in bioluminescence. He wanted to see some of the bioluminescent specimens that we had, or specimens of animals that are bioluminescent when alive, and he wanted to come and watch me work. And I didn't know anything about the program. It was brand new, and I got this email. Of course, my first reaction was, no way. I don't want to have somebody come in and to sort of watch, or have somebody who was an artist who was going to start looking at our specimens and what damage could be done to them if we didn't really pay attention. But the Smithsonian being the place that it is, we are a cooperative group, and when something like this happens, we usually go to the next step. I thought, well, let me bring him into the museum and I'll talk to him. And so this is CJ in the museum. He had come in and he wanted to look at some specimens. And what was really wonderful about it is that he was very excited because he said he had only seen these in videotapes. He knew he had tried to study bioluminescence and bioluminescent organisms and how they're put together, but he was never able to, he didn't even conceive of having the chance to actually touch the specimens. And so here he is, he's examining some specimens of some deep sea fishes. I just pulled out one on the left here, one of the deep sea anglerfishes that bioluminescent bacteria take up residence in this the modified first dorsal fin spine, and these, when preserved, well, at depth, say at like 1,000 meters, they'll be all blown out. But when a preserved specimen like this is really sort of bizarre. It feels like a water balloon. It's a very strange thing. But anyway, what the important part about this is that CJ was very excited about looking at the specimens. He treated them with respect and with awe, and spoke about them in a very nice way. So I thought, well, this is great. He can stay. And other people within the museum, educators and people in exhibits, had also caught wind of his work in the museum, and we did some public programs. I'll talk about some of those with another artist in just a minute. And so we had just a few years before this had opened our new Sant Ocean Hall on the main level of the Natural History Museum, and it was really quite a spectacular exhibit. And it includes at one end a temporary exhibit hall, and CJ, working with myself and exhibits and education people, put together an installation called The Bright Beneath. I wanted to call it Light at the Museum, but nobody wanted that one. So, so the luminous art of C.J. Huang. It ran for a little over 5 months, from the end of September into early January. It was very well attended. Again, we had some other public programs that went along with it as well. Okay. And this is what one of his installations looks like. This is not the one at Natural History. I didn't have a good photograph of really the whole thing. And so this is one, I think this was in Australia, but when it's up and running, these are all, say this plastic tube here will fill with air and then deflate, and these actually look like organisms, not recreations of organisms, but just his interpretations of them. They're really spectacular, and you can see his work at messymix.com. He's really an effective communicator, because you can all remember that, I think. Yeah. So that's the SARF program is up and running, and it's becoming more and more popular. So I took another SARF fellow, and this was a woman, Annette Kauenberg, who was a fiber artist at MICA. And MICA is the Maryland Institute College of Art in Baltimore. It's one of our premier art colleges, on a par with Pratt Institute and so on. And she's been associated with me for a few years, and we just finished a program earlier this year. I'll talk about in just a second. This is, we were at a, so she had come in to work with me. She was very much interested in clothing and how organisms are covered or protected and so on. And we had a public forum on a Sunday afternoon at the museum, and people were very excited about that, to see scientists and artists together talking about their work. Her project was called The Skeleton and the Skin, and this is a real specimen. This is a sturgeon skin and its scutes. And this is some of her, one of her pieces of Dutch collars. She's originally from the Netherlands, and she was very interested in this, in the form and the repetition of form, and got very excited about the scutes and how they were put together as well. she loved the microscope, and I'm convinced if you just put a fine artist in front of a microscope, that you'll get a lot of interest and attention within the museum. Now she's looking at some histological slides. CJ had looked at some whole organisms, so she looked at the histology, and that she really loved. She loved the whole idea that you could take a specimen and chop it up and then look at the little pieces. So this is again an ancient technique, but we use routinely at the museum still. And she put together her installation, which was at Mica. And this is a long, maybe about, 6 to 7 foot long light box. And it's got, she took, I was really sort of stunned to see this. She took, these are images, histological slides, sections through some gobies, and they're laser cut, so they're burned onto this buckram material, which is a material that's used in making hats, and then she lit it behind with a light box. So again, it's not scientific at all. This is completely art. And one of the things that surprised me about these kinds of things was that they attract attention. There was an article in the Science and Culture section of PNAS just on April 12th. It's entitled "How Fish Innards Inspire Art." Not the title I would have picked, but I think it's okay to get two full pages here in PNAS. This is going out to scientists of all stripes and ones who would be affected by this and understand that there's something is going on within natural history museums that perhaps they didn't know anything about. Here, this is one of her pieces. It's an origami piece that was inspired by fish bones and skin and how they're put together. She makes some bigger ones as well. This one was probably maybe about 2 feet wide. She has some more elaborate constructions that she's working on right now. And then finally, the Sturgeon Plate really grabbed her attention, and this is we went to the College Art Association SARF Forum. This was a College Art Association. It's a big meeting of students and faculty from around the country. It was in Washington, D.C. in February, and this is a real sturgeon plate mounted, and she made a new one, mounted it. But the way she made it was she did a 3D image of this one, and then she took it and she made 505 slices out of buckram and then put them all together again. And she was just interested. She said, if you take a specimen and you slice it up and put it in pieces, I want to make one and then put it back together again. I mean, it's a funny concept, but it's an artist. So, but again, the point was it gets attention. It's a vibrant program. It opens us up to a wide range of people who had no idea that we even have specimens in the Natural History Museum, and I think it's been a lot of fun. One of the things that people have asked me is, has my research been affected by having an artist working with me? And so far, I haven't done any project that's come directly out of this, but we've been talking about, we have a lot of histological slides, and possibly we have some of the brain of a coelacanth, and we're thinking of maybe scanning those and doing some sort of 3D reconstruction of those. So that's something in the future, but we found it's a lot of labor intensive. Just a scanning of these slides gives us huge files and so on. I know that the capability is there, but we haven't gotten to that yet. So that's possible to do in the future. If I do it, it would probably be something that's more, I told her it's going to be more research than art, because what I told you so far, I get one little bit of credit during my annual performance appraisal. I'm in the museum to do research on fishes, not to talk to artists, but this is an important thing, I think, to do, and it opens us wide open to lots of different opportunities. Okay, so that part is about inviting artists to study natural history specimens. I now want to talk about mining old collections for new data. Now, this is something that somebody who's a systematist, comparative anatomist, does all the time. You know that if you, the more you know about some specimens, the more you can know if you just do a CAT scan or you do some SEM and so on, just go dig deeper and deeper. But the point I want to make about these is I want to talk about some projects that I've been doing in reproductive biology of fishes. And I use reproductive characters in a systematic way in the groups that I work on, usually looking for diagnostic characters, sometimes of gonads for taxa. And this is one project here. This is a deep sea fish, Chlorophthalmus agassii. And this is from one collection that was taken off Florida in June of 1958, so almost 60 years ago. It's a hermaphroditic species. We know a lot of deep sea fishes are, and its range is from about 50 to 1,000 meters. And so the work that I've been doing in collaboration with the Florida Marine Research Institute and specifically a colleague of mine there named Harry Greer is to, when research biologists at a marine lab want to look at say the reproductive biology of a species, they think they have to go out and collect them. They have to collect them every week, every month, or something like that over the period of a year in order to get some understanding of their reproductive biology. The thing about that is you're not going to be able to get, unless it's a very common food or sport fish, you're not going to be able to get a lot of the specimens. And imagine if you wanted to study the reproductive biology of the deep sea fish. I mean, how do you do that? How do you go out and get specimens all the time? And so what I've been and successfully convinced Harry and some other colleagues that we have specimens within the collection that you can use that would be useful for studying reproductive biology. And just for this one particular project, we took just 10 specimens from this collection and prepared histological sections of them. Okay, we could immediately determine that it has a hermaphroditic gonad. This is a section through the gonad of one specimen. This bluish portion here is the testicular portion, and there is sperm in the testis lumen, and the darker purpley or reddish purple part is the female portion of the gonad. And these are in a more primitive state of primary growth oocytes, so that's not mature oocytes. So this specimen is alternating between male and female, we infer, and this particular specimen was in the male phase at that time. Another specimen here is an ovulating individual. It's difficult to say it's either male or female because it's usually both during the lifetime. And here this is quite an interesting section. In this one we see oocytes in various stages of development. And I'll just go through some of those quickly. Now here, these are primary growth oocytes. These are in the earliest stages of the development of the oocytes within the ovary. We have a secondary growth oocyte, one here and one here. Secondary growth within the development of oocytes it means the onset of vitellogenesis, which is the formation of yolk. So to go from primary growth to the secondary growth, yolk granules are formed. That's what these are right here. And then we can also see in here POF is a post-ovulatory follicle, and so this follicle had an oocyte in it before, but it has been released, so that the specimen has spawned that oocyte. And so we know just from this section alone, it's so detailed and gives us so much information that we know that this species is an asynchronous spawner, which means that it's spawning. We infer sort of continuously throughout the reproductive season, as opposed to, say, a synchronous spawner, which would be something like a salmon, which spawns just in one go. That's one difference that we make. So we're learning a lot about this. Again, this is just 10 specimens collected 60 years ago, sitting in our collection. Okay, here's an ovulating individual, and this is an oocyte just prior to ovulation. These are yolk granules that start to come together. They will coalesce. The germinal vesicle or the nucleus moves to the side of the egg when it's going to be fertilized, and this is just that same portion, a little larger. And as you can see here, there's a micropyle and that's what fish eggs have micropiles. Fish sperm don't have acrosomes, so there's a hole in the egg through which the sperm go. It's very difficult to see a micropile. We don't have documentation of micropiles in very many fishes, as you might imagine. I mean, we know about it in the fishes that are used commonly within fisheries biology for food and so on, but not in something like this. And looking for a micropile is like looking for a needle in a haystack. So we're really, quite excited to see something like this. And then finally, the last section that I want to show you from this is these are cells that were caught in the mitotic cell division of oogonia. So here's a prophase, oogonium, anaphase, and metaphase. And so the point is, and Harry really took this on, is that there's a lot of information that you can get about reproductive biology from our collections, and all you have to do is look for it and use them in a different way. That is, for a fisheries biologist, this is a different way to look at collections. And sometimes when I talk to paleontologists, they say, after when the biodiversity crisis comes to a terrible end, everybody will become a paleontologist because there'll be nothing else to study. But I think what people do is become comparative morphologists, because we have just in our museum alone about 5 million fish specimens, and we don't know this detail of reproduction on very many of them at all. So I think that's some little reason to be optimistic, I suppose. Okay, the last topic that I want to cover is the cryopreservation of living cells, and this is an example of making new collections, adding new collections, new types of collections to the Natural History Museum. And as many of you know, we have what we call the Pan-Smithsonian Cryo Initiative, and this is just the different units throughout the Smithsonian in which frozen specimens are collected and maintained. Of course, the zoo is prominent in that work, the Natural History Museum, the Smithsonian Environmental Research Center, and then, of course, the Smithsonian Tropical Research Institute in Panama. And Piper Mullins, I have her name on the lower left here, is one of the coordinators of this. She's based at the zoo. And we had a workshop in 2012 about the possibilities and the options for developing frozen tissue collections, and she gave us the data that at that time we had about 250,000 samples, with a capacity at our new facility to have 2 to 4 million. Those of you who are interested in these kinds of collections, when you come to Washington, we should take you out to the facility. It's pretty spectacular. We just 58 freezers, 24 nitrogen tanks. I mean, it's really quite a lot of effort was expended to build these collections. We have all vertebrate tax groups are represented, and then insects, plants, and then some ecological samples. But these are what we don't have in natural history, is cryopreservation of living cells. All these specimens so far, or at least in 2012, were of frozen tissues, where there could be, say, a fin clip or a piece of muscle that was dissected from a specimen and put in liquid nitrogen and stored at minus 80 degrees centigrade. That was it. That's not a living cell. That's tissue that can be used for sequencing or for some other possibilities, but not for. It's not living tissue. And I became interested in living tissue. I'm not a cell biologist at all. Get that clear. But I became interested in expanding the collections and getting cryopreserved living tissues into the museum when I started doing some work with Mary Hagedorn on the left here. Some of you may be familiar with her work. She's very prominent now in the cryopreservation of stem cells of corals, and is off to Moorea this weekend actually to participate in some preservation projects there. I have this title up here, Marine Geo Hawaii. Marine Geo is a project in the Smithsonian that is focusing on different labs and areas throughout marine habitats in order to set up some systematic assessment and monitoring programs. Mary is the director of Marine Geo Hawaii, which is based in Kaneohe Bay on Oahu. Mary is very enthusiastic and likes to go out and tell people how easy it is to cryopreserve cells. I'm the one with the net getting the fish. I don't do that. Really a collaborator in the strictest sense of that word, but she's the one who's the cryobiologist. One of the reasons we talked about this, we told people, why don't you collect cryopreserved material, and basically they said, they don't know how to do it. They don't know how to do it in the field. It's too difficult, and so on. And so we thought, well, we have to make it easier again. We can't say, okay, you're right. We have to try and get some method together. Cryopreservation of living cells differs from freezing cells for freezing tissue for molecular biology, of course, in that you have to create a cell that is basically frozen in sort of what we call a suspended animation. You have to take water out, you have to put cryoprotectant in. Water comes out of the cell very readily when you freeze it. I mean, everybody knows that when you put food in your freezer and you come back, a few days later, there's ice crystals. It's because it froze too quickly. It's fine for your food purposes, but if you did that for cryopreservation, the cells would be destroyed. So we had a project in Hawaii where we used some Danio, which is a common experimental fish, and a goby that was native to Kaneohe Bay. This is commonly known as a zebrafish. This is a starry goby. The zebrafish were purchased commercially. They don't live in Hawaii. And we got together put together what we call a practical method to cryopreserve fish stem cells. And so what you would do is dissect the testes out, put them in a phosphate buffer saline that's also been diluted with bovine serum albumin. This is sort of a typical procedure that's done for cryopreservation of cells elsewhere. There's a lot of work, of course, that's been done in human reproductive biology, so you don't have to reinvent the wheel for any of this. Homogenize each sample, count the cells, and that was done for testing to see how well the process had worked. Each sample is diluted again, fill the diluted sample in cryovials. These are the ones in which they will reside until they're used. Cryovials that are placed in a passive freezer, and then so there's some ice in there, and then they go put into a minus 80-degree freezer for 4 to 8 hours to freeze at 1 centimeter per minute. And so that's where the idea comes in, that this has to be done slowly. You can't freeze them right away. It has to be a slower process. And so as they freeze, as the water comes out, the cryoprotectant will go in. And then they get immersed in minus 196 degrees centigrade until you're ready to use them. And when we spoke to some people about this, they said, I can't do this in the field. I won't be able to take all this stuff with me. But I disagree. I think that 20 years ago, people said they weren't going to take liquid nitrogen and dewars and other equipment into the field. They do that now readily. And I think if people want to do this, they will. But we're not going to push it too much. Initially, we'll start at these marine labs, so that we'll develop the collections, and so people can get an idea of how those work. So I'm happy to say that now we have some cryopreserved testicular stem cells of this goby in the Natural History Museum. They're stored at minus 196 degrees centigrade and are available for use. And the uses of these cells are many and varied. Of course, they can be used to regenerate or recreate populations. That's one of the most exciting uses of them. And then also, of course, it gives you sort of gold standard sequence data. So just to conclude, these are the 3 ways in which I'm personally involved in some efforts at the museum to expand the use of natural history collections, to bring them more to the fore, to let people know about how valuable they are. And I think my view towards this is that I'm looking really ahead to future generations who will look back at us and say, you knew how to do this. Why didn't you? So why not? Thanks.